discovering the structure of DNA really opened up the gates to all genetic research that's happened after that, because by looking at the structure of DNA and working out where the molecules fit together, we can also work out key issues like how DNA is replicated, and then once we know all about the enzymes that do transcription, we can also work on the structure of DNA to unravel the secrets of that. So Watson and Crick's finding or discovery of the structure of DNA was pretty well crucial to the kind of biotechnology that we have now and to our understanding of what goes on with our DNA and the production of proteins in our cells. The story perhaps started with Oswald Avery's discovery of the transforming factor. The transforming factor was what one bacterium could transfer to another bacterium, and then that bacterium would have a different gene. It would have a different genetically encoded ability from what it had before. So this must be the genetic material, because if it takes one gene from one organism and puts it in another organism and gives it the genetically encoded ability, then that is the genetic material. Avery then showed that this genetic material could be destroyed by a DNase, but it could not be destroyed by a protease. And this was absolutely crucial, because before that, people knew that chromosomes were the things that transferred genetic abilities from person to person, but the chromosomes were a combination of DNA and proteins. So the genetic material must be DNA or protein. Most people tended to favour proteins because there are many, many kinds of proteins, and the complexity of the number of proteins seemed to approach the sort of number of genes that exist, or the number of characteristics that were passed on genetically. People didn't really consider DNA as much of a candidate because it only had 4 bases, and 4 bases just did not seem intuitively enough to carry all of that genetic information. But Avery showed that it was, by showing that the transforming factor, which did convey the genetic ability from one bacterium to another, could be destroyed by DNase and not by protease. Clearly, the genetic material was DNA. Well, after that, there was an explosion in people studying genetics. And in order to do that, one of the greatest tools was X-ray crystallography. Now, people studied X-ray crystallography at the Cavendish laboratories in Cambridge and also at King's College laboratories in London. Also in California at the same time, Linus Pauling had a very good understanding of this technique of studying molecules. So what they did with the molecules was they crystallized them. So they removed the water, just like you'd crystallize sucrose, sugar, by removing the water, and it forms crystals. DNA can be studied in exactly the same way, by drying it out, making a crystal, and then shining a ray of X-ray through that and working out the pattern that is made when the X-ray is diffracted by the crystal. Now, the person who really comes to the fore here is Rosalind Franklin at King's College in London, working in Maurice Wilkins' laboratory. And she studied DNA, and she discovered that DNA was a symmetrical molecule, and also an analysis of the coordinates on the points indicated by the photograph that she took of a crystallogram of DNA showed that it was probably helical. And this was kind of interesting because it relied on very, very precise interpretation of the photograph, and someone who wasn't trained in crystallography could do that roughly, but could never do it as well as someone who was really good at it, such as Franklin or indeed Linus Pauling working at the same time. So she got a rough idea of the symmetrical nature, which indicated maybe 2 strands to later workers, although that wasn't obvious at first. And she also got the idea of the 2 nanometer diameter. So we go on to Pauling, who looked at some of his crystallography photographs, and his actually weren't as good as Franklin's, and he didn't get that diameter indicated to him very clearly, and he eventually produced a kind of common sense model of DNA. So it had 3 strands, the phosphate sugar backbones, in the middle, and it made sense to him and to pretty well everybody to put the bases on the outside, because if the bases were accessible to enzymes, then they could be read and you could make proteins based on that genetic code. So Watson and Crick also made a model slightly before Wilkins, and it had the same errors on it. But that never got published, so Pauling never found out about it. The reason it didn't get published is that Wilkins and Franklin came down to London to examine their model, and it was based on Franklin's work, but Watson had misinterpreted the work and in fact misheard and misremembered some of the key statistics. So when they presented their model to Franklin, she ridiculed it and it never got published. Pauling never found out about this, so he published his model and received a bit of low-key criticism as well, but not quite so much ridicule as the young Watson and Crick got. So then, once everyone could examine that model, everyone realised the shortcomings of it. In the meantime, Erwin Chargaff was doing some painstaking, long-taking chemical analysis of DNA, and he found that the number of molecules of adenine equaled the number of molecules of thymine, and the number of molecules of cytosine equaled the number of molecules of guanine. And that was true in every organism he studied, which was a lot. Nobody knew how to use this information, and it was Watson and Crick maybe who made the crucial discovery that we could link that to the structure of DNA, especially when we looked at that 2 nanometer diameter and the helical structure that Franklin had come up with. In addition, she'd mentioned a symmetrical format, and the symmetrical format corresponded to 2 strands. And the final piece of the puzzle really came when Watson went into the lab and played about with cardboard cutouts. And the cardboard cutouts had exactly the right angles of all of the different atoms on the A, G, T, and C bases. And he knew this idea of 2 nanometers, and he put a purine, which was a 2-ring base, such as guanine and adenine, together with the 1-ring base, the pyrimidine, such as thymine and cytosine, and he discovered that they would fit into quite a nice shape and size. And if you added the diameters of the phosphate sugar backbones, that gave him a molecule of 2 nanometers diameter. The last thing he had to do was fit those together. And he kind of accidentally flipped one of the cardboard models upside down and discovered that they did indeed fit like this. So only if he had the models anti-parallel could the various parts of the bases actually be in the correct alignment where they would form the hydrogen bonds that Watson now realized had to hold the DNA molecule together. And so that combination of X-ray crystallography giving us various vague details, Chargaff's work of the equivalence of A and T and C and G, and a whole lot of people working together and making mistakes and then coming back together and amending those mistakes led to Watson and Crick actually getting the credit for this. After all, they did make the crucial intuitive discovery of the anti-parallel nature and therefore the hydrogen bonds holding the DNA molecule together. And then after 1953, once people knew what the structure of DNA was, suddenly the world was open to the study of genetics, and people could work out how it was replicated, how it could be transcribed, and later how it could be manipulated to form the study of biogenetics and genetic manipulation that we take for granted now.